What we're going to do now is we're going to use a Pasteur pipette to take some of this culture and to place it onto this nutrient agar plate. And then to get a nice thin film of microbes over the surface, we're going to use a spreader. But of course, like all microbiology, we have to adhere to good aseptic technique. The first thing we need is the Bunsen burner. So if you turn on the gas, then again we're going to have a nice upward draft so that we've got a nice working area. In here I've got a number of Pasteur pipettes. What I'm going to do is to open the end so I can take one out. I find it easier often to use a pair of scissors just to cut the end off. I've got a technique of holding 4 of them and letting the last one drop out into my hand. When I've got it there, all I'm going to do is to place the teat pipette onto the end of like that. I don't take it out too far. I've kept all of that protected inside the container. Because I've still got 4 that are sterile, I'm going to try and protect them. So what I will do is I will take that out, fold that over and protect them so I can use them again. And I'm going to use a method to hold the Pasteur pipette like that. I will check the top is not on too tight, and all I'm going to do now is this. I will hold the top in my little finger and turn the bottle. Flame the neck, apply a little pressure to the teat. And because I'm holding the glass, I cannot put inadvertently pressure onto the rubber teat. I flame the neck and return the top. I try to make sure that I always keep my arm fairly stable so that I don't drop liquid culture over the bench. I lift the lid and I'm going to put 1, 2 drops, just applying a little bit of pressure to the teat. Then I put the lid back on. I'll move the disinfectant a little closer to me, place the teat, take the teat off the Pasteur and let it drop in. And that's all there is to it. Now what we want to try and do is to spread that culture all the way over the agar plate. I've got a sterile plastic spreader. I open this end, take out the spreader like that, and now I can gently move that culture all the way over. I've got a little bit of a technique here of being able to kind of pull the plate round and just gently rotate it. If you're not so sure about that, then just gently move it all the way over like that. The thing that I don't like to see is people putting their hands over the top, because there is the possibility that cells or bacteria could drop onto the plate. So if possible, try and keep your hands to the side. When you've got a nice thin film all the way over, put the lid back on there, and then I can drop the spreader into the disinfectant. That's all there is to it. Now I thought it would be nice for you to have a go. Only this time I used a disposable plastic sterile spreader. You've got a glass one there that can be reused. Okay. It's all yours. Off you go. Oh, you'd like the scissors. Here you are. There's the scissors.
Right. If I just hold on.
Yeah, hold on to the 4 and let one of them come out. Yeah.
Okay. And then just a couple of drops?
Yeah, 2 drops will be ideal.
Okay. Okay. Now, the glass spreader, the same technique?
Same technique, we'll cut the end off.
Okay.
Remembering to open that end and not the other end.
Okay. then just place that in there.
Place that in
too.
there. And the other thing we'd do is we'd leave this for a little while just to let the liquid soak into the nutrient agar
Yeah.
so that we don't get a lot of liquid floating across the top.